Hi everyone, thank you very much for joining us for today's announcement video. We are talking today about our new Ataraxis Tau platform and the first test that we're launching built using Ataraxis Tau platform, which is our test to predict adjuvant chemotherapy benefit. I'm here today with two of our research scientists, with Linus and with Ken, and we're excited to tell you about our breakthroughs and advancements and what we've accomplished over the last couple of months. Can you guys introduce yourselves?
Hi, I'm Linus Bao. I'm an AI research engineer at Ataraxis AI. Prior to Ataraxis, I graduated with a master's in computer science from Oxford University. And at Ataraxis, I focused on graph neural networks for digital pathology and causal inference.
Hi, my name is Ken Zhang. I'm a research engineer at Ataraxis AI, and previously, prior to Ataraxis AI, I was a researcher at N1U Langone working with Jan here on predicting patient outcome.
Great. So guys, can we maybe first talk about what even is Ataraxis Tau platform? You've been working on this for the last couple of months. Can you tell everybody in simple words, what is Tau and what is the adjuvant chemotherapy benefit prediction?
Yeah, sure. So essentially what we're hoping to do is, given a breast cancer patient and their clinical characteristics, characteristics of their particular histopathology, we hope to predict the individual treatment benefit that they would experience if given chemotherapy. And we do this using a deep learning model trained on retrospective observational data. So this gives us a lot more data to work with than if we were restricting ourselves just to clinical data. And sort of the unique thing about what we do is that applying deep learning methods to observational retrospective data is it's really at the forefront of machine learning and causal inference research.
Traditionally, a lot of these tools have been developed using some clinical trial data, which means these are very curated set of patients, treatment is randomized, they are followed up for a long period of time. Whereas for us, right, we're trying to develop these from what we call observational data, which is just patients collected from hospitals around the world. And so the downside of doing this is, right, is that let's say you train a naive model, your model would learn quickly that, oh, patients who are given chemotherapy generally has worse outcomes, so you should never apply chemotherapy. But we know this is not true because in the real world, there's something called a treatment assignment bias. This means that patients who are generally higher risk tend to be more likely to be given chemo. So essentially, we have to modify our models in order to adjust for this bias.
Can you talk about the causal inference part of it? So before causal inference work at Ataraxis, we've worked a ton on just predicting patient outcomes, predicting risk of recurrence, and applying those methods to digital pathology imaging, to clinical data. But now the work is moving towards causal inference and looking at different treatment effects. So what is different? How is this different from just predicting a risk of recurrence?
Yeah, so sort of the fundamental issue in causal inference learning is that we don't have full access to the quantity that we're trying to predict. So what I mean by that is we're interested in understanding the benefit of chemotherapy for a particular patient. What does that actually mean? We look at a particular patient and then we ask the question, what are their outcomes if we don't treat them with chemotherapy? Then we also ask the question, in an alternate universe where this exact same patient does receive chemotherapy, what are their outcomes? Now, of course, the issue is in the real world, we only see this patient either receive chemotherapy or not receive chemotherapy. We can't really peer into this alternate universe of what if they had gotten the other option. So the closest we can get in this case is clinical trial data. But the issue with that is that clinical trial data is relatively scarce compared to retrospective observational data. So solving this problem really necessitates getting very creative with how we train our models. And we need to sort of leverage the partial information provided by observational data in order to sort of simulate a clinical trial.
I want to say that it's been a breakthrough because this idea that you can just look at real world data and you can infer the treatment effect or recover the treatment effect. It's something that people have been trying to do for, I guess, decades. And especially now with a ton of data, with new methodologies, we are able to do it. And starting with adjuvant chemotherapy as the first application of those causal methodology. And can you maybe tell us how does it work? So when we start working on those methods, like what gets us that breakthrough, and what kind of work have we done to get to this moment where we actually can look at real world data and predict patient effect?
Yeah, so the biggest difference between an observational data and randomized trial is that in the observational data, treatment is assigned non-randomly, whereas in a randomized trial, everyone has an equal chance of getting treatment. And because of that, I think it's much more easier to use randomized trial to draw conclusions about the effects of treatment. So essentially, in our work, we have to take observational data and try to simulate that as a randomized trial to be able to train better models. And we do so through a process called adjustment. So essentially, that means that we have to weigh different patients differently based on their characteristics, such that overall, this very uneven distribution of treatment effects becomes more even, like a randomized trial.
Okay, so why hasn't it worked in the past, and what have you done at Ataraxis that suddenly it starts to work? Is it a combination of data, completely new methods? What brought us to this point that it suddenly starts to work?
Yeah, so Ataraxis has done an amazing job at collecting data from across the world, from 20 plus different hospitals. I don't think anyone else has made such a huge effort to collect so many patients from across the world. I think previous studies usually focus on one particular cohort from one particular hospital, or even just like 3 cohorts, whereas we have like 20.
Yeah.
So I think that data collection effort really made this possible.
Yeah, we have a ton of partnerships with different health systems in the US and around the world. To our knowledge, I think we have the largest collection of real-world data that includes digital pathology, but also long-term outcomes. So we actually know what did that cancer look like 10 years ago, what kind of treatment the patients received, and then what happened to those patients. Now, that's one part of the equation, because you still need to recover that actual treatment effect and know what causes those patients to benefit from any particular type of therapy. But we had a lot of the ingredients to do this. And also, Linus, is there something special about the methodology, like knowing that you are dealing with data from multiple institutions, and it's not just one trial or it's not one site, but it's a mix of data sets from different institutions around the world? Is there something that makes it more difficult? I know it makes it more difficult to build those models, but how did we end up working with this kind of data?
So when you have data all from a single source, you can make a lot of simplifying assumptions about just the types of techniques you can employ, the types of things you can do with this data. When you have data from all over the world, 20 different cohorts, you have to get kind of creative with how you mix information from different cohorts, and it sort of requires you to, I mean, without getting into too many technical details, it sort of requires you to evaluate your models in a different way, to think about adjustment in a different way, because as Ken said, the main issue with adjustment is that you want to simulate randomized treatment assignment from treatment assignment that isn't random in observational data. And when you have so many different data sources, the non-randomness of your treatment assignment is different across different data sources. So of course, adjustment gets a little bit difficult. Everything just gets a little bit more complicated, but if you're able to leverage multiple cohorts correctly, which I think we are able to do that, then you get much better outcomes than if you were just training on a single cohort.
Yeah. How is our chemotherapy benefit prediction different from already existing assays that are prognostic tests, but make a claim that you can identify patients who benefit from chemotherapy?
Yeah, so today the standard of care in chemotherapy is something called Oncotype DX. It's a test that predicts your chance of recurrence within the next, I think, 10 years. And the claim they're making is that patients who are higher risk tend to benefit from chemotherapy. And this makes sense because if you have a more aggressive cancer, you should be getting more aggressive treatments. However, the converse is also true. Even if your cancer is more aggressive, doesn't necessarily mean that you're more likely to benefit. So our method's very different from their approach in that for each patient, we not only predict a single risk score, we predict the risk given 2 different lines of treatment. This makes it possible to have patients who are generally high risk, but we might show that they don't actually benefit from treatment. And the reverse is also true. We might have low risk patients who can benefit a lot from treatment.
I think randomized control trials are this perfect example when you are introducing new drugs. And I think typically clinical trials have been designed, to introduce new drugs to standard of care. And you can get a result, that whether the treatment worked or not. Now, if you want to build a tool that can predict treatment effect, you kind of have to use this data because it's closest to an unbiased assignment of who is getting what. And that's what you kind of need if you want to fish out the right features that are associated with treatment response. You just pair what you saw at the baseline, you pair some clinical characteristic or something in histopathology or molecular data, and you try to pair it to patients who had recurrence, who survived, whatever endpoint you're interested in. You match it to characteristics at baseline, and that is ideal world, but access to clinical trials is difficult. There's much more data in the real world. So is it possible to use both? I think so far we've spent most of our time on using real world data. And how, so if we had access to clinical trials, we are doing that for evaluating our tests. And we are applying our tests to show that in an independent validation, we can predict chemotherapy benefit or other therapy benefit. But also, is there a way to combine information from real world data and clinical trial data? And how do those two differ? Because can you apply the same methodology to recover the effect from real world data and controlled trial? Can you also?
Yeah, so typically the methodology we've designed is particularly for real world data because what we're trying to do is we're trying to reduce this bias in the treatment assignment that you've spoken of. So essentially, just to give a high level overview, the main issue with data that doesn't come from a clinical trial is that, let's say you want to know the effect of chemotherapy, very naively you can say, okay, let's look at the average health outcome for those patients that did receive chemotherapy. How does that compare to the average health outcome of those patients that didn't receive chemotherapy? If you did this on real-world data where treatment wasn't randomized, you might actually see that patients that receive chemotherapy are worse off on average than patients that did.
That's what's happening, right?
Yeah.
If you look at every data set, you're going to see patients who have more aggressive cancer, they recur more often, even though they received chemotherapy.
So there's sort of like a reverse causation here. You might think, okay, is chemotherapy making these people's cancers worse? But of course, that's not the case. Really, it's that doctors give chemotherapy to those patients that were worse off in the first place. So typically what we do when we train on this type of data is we need to do some adjustment, some shuffling around of these distributions, the distribution of treated patients, the distribution of untreated patients, such that we can eliminate this particular bias in the data set. Now, if we were to also train on clinical trial data, we would treat this data differently because this bias is not present in the first place. So there surely, I think, in theory, is a way to leverage both types of data, but the way that you would do it, you have to sort of design the way you're training a little bit differently if you were to do this.
All right, let's spend a few minutes talking about that. How do you do it? How do you make sure that you can do it from real world data?
Yeah, so there's a lot of different approaches in the literature, and at a high level, essentially what you want to do is take your biased real world data and sort of reshape it to look somewhat like clinical trial data if you squint. And it's sort of a messy thing. There's a lot of sort of like little engineering tricks that you have to do to make this work that maybe aren't the cleanest in a theoretical setting, but they give us the results we want.
Okay, so what is it like the data, the amount of data that made it possible, or the methodologies? What is this?
Yeah, so recently there has been a lot more work done in designing novel objectives that allows us to train these causal models from observational data. And secondly, right, deep learning has been huge recently. Allows us to like optimize any objective we want on any data with like any sort of input sizes.
Can you explain what does it mean that you have different objectives when you're training those models?
Yeah, so essentially, if you were to train a naive model and just like try to optimize whether the model predicts what actually occurs, the factual observation, you would end up with some models that's similar to what Linus said. Essentially, it just predicts that if you give them chemotherapy, the patient will be worse off. Mm-hmm. We know that's not true. So we have to change our objective in order to not only minimize what is factually observed, but also what we think is happening in the alternate reality where the patient was given a different line of treatment. A different way of thinking about this is essentially one objective that eliminates the sort of bias caused by treatment.
No, I mean, yeah, it's a really difficult thing. There's a lot of alternate perspectives that you can take on the problem, whether it's matching the distribution between treated and untreated patients, whether it's doing that or sort of reweighting certain patients to be more influential to the training objective, or whether it's having some sort of explicit constraints on the training objective, which avoids solutions that go against our prior knowledge on the way treatment should affect patients.
Yeah, or using prior knowledge where there are certain groups of patients where you know definitely that they would benefit, and forcing the model to predict the positive benefit for these patients.
And you know that from old clinical trials or whatever in literature, and you can predefine almost those patient groups where you're expecting treatment benefit?
Yeah, we learned a lot of these from existing clinical trials, and also I think just from our intuition about how chemotherapy works.
Yeah. It's a subtle thing because we want our model to be concordant with things that we're very, very confident to be true in terms of what subgroups we expect treatment benefit in and whatnot. But we also don't want to just recreate just regurgitate the knowledge we already have from clinical trials. We need our model to go a step further and tell us something that we didn't know. And that kind of touches on a whole another question of evaluation is once your model does make a prediction, which is maybe not something that's already known in the literature, how do you evaluate it to be true? This is a whole nother can of worms. That's a major difficulty in causal inference research in general, but it's something that we're working really hard to figure out.
So far, the approach is you just look at the risk of recurrence, and the hypothesis is if you have higher risk of recurrence, you would benefit from more aggressive therapy, which on average is true, and it's been shown in clinical trials that it works on average, and vice versa is true as well. Patients with low risk of recurrence don't benefit from aggressive therapies, but it's a bit trivial approach that right now there's just no way that every high-risk patient benefit from adjuvant chemotherapy. And what we're saying is that, yeah, there are high-risk patients who don't benefit from adjuvant chemotherapy, and vice versa. There are low-risk patients at low risk of recurrence, where you can further reduce this risk if you apply adjuvant chemotherapy. But I think it's done for the first time. Have you guys seen any examples of this? Maybe in literature, but not in the real world.
Yeah, most of these causal analysis models, they're done using synthetic data. What that means is that the researcher design what the data is going to be, and then they train their model and show that it's able to predict the thing that they design. Where for us, right, we're trying to answer a very open-ended question. We don't know the answers, and which is why it's so difficult.
Yeah, but I think, Jan, what you've said really gets at the heart of the utility of what we're doing is we're sort of moving beyond the average, right? Clinical trial, classical literature on the topic gives us a lot of knowledge about, okay, maybe this group or that group on average benefits from treatment, but there are, of course, people within these subgroups that deviate from the average. The average is, of course, like a reductive metric in some sense, right? So sort of what we want to do is we want to be able to say this particular patient, their particular unique characteristics, do they benefit from treatment or not, regardless of what subgroups they fall into.
The other thing that we are moving away from is, in the past, if you wanted to do something like this on pathology slides, then you would go to 100 pathologists and ask them to essentially try to identify if there are specific cell types, maybe structures, how they organize that are associated with outcomes. And that was the entire idea. And we have some features like tumor-infiltrating lymphocytes that have been found to be associated with outcomes, maybe even with immunotherapy response. But it's a bit futile to find one very particular feature and hope that it's going to be associated with outcomes, especially when there's just so much data in the pathology side. And those images are so high dimensional, there's so much information that I think we've started to move away from just looking for one very specific feature and try to find hundreds of features or tens of features that might be associated with outcome.
Yeah, because when you think about it, right, there's like hundreds of treatments, there's like tens of different endpoints. It's just simply not possible to identify a feature that corresponds to benefit to this very specific line of treatment. Rather than that, we're hoping that we can train models that automatically figure out which features are important. And one of the ways we do that is we have a foundation model that, rather than picks out specific human known features, it generates embeddings that encode what the information is in the slide. And based on these encodings, we can then train models to predict anything you want, from recurrence outcome, chemotherapy benefit, endocrine treatment benefit, etc.
Yeah, and the key is here, of course, like these foundation models are trained on such massive amounts of data, more data than a human pathologist would see in their lifetime. So of course you can pick up on much cellular patterns in the data that a pathologist could never pick out just looking at things manually. And we can sort of cater them very, very nicely to whatever objective we're interested in based on the flexibility that deep learning provides us in objective functions, as Ken has mentioned before.
Yeah, and for whatever, I guess also treatment. So if in the future we collect an order of magnitude more data, and that data is going to start to contain information about therapies that were relatively rare, but we're going to have enough information to even apply it to new types of therapies. So now we're moving away from adjuvant chemo benefit prediction to whatever endocrine targeted immunotherapy there is, right? The platform is not necessarily tied to any specific type of therapy, or maybe not even specific to breast cancer at all.
Yeah, for sure. I mean, our general methodology is just, it's something you can really apply to any causal inference problem. So any problem of estimating treatment effects based on observational data, we can apply the same thing we've done in the chemotherapy setting. We're interested in chemo as a first step just because it's such a popular treatment option, but we definitely are working to move beyond that.
I'm excited about a lot of things. We're going to be looking at clinical trial data to evaluate those models, to improve on them. We're moving beyond adjuvant chemo benefit. What gets you excited from the scientific part, and what are you looking forward in the next few years as we develop the TAL platform? Because this is just the first step, really, and we're showing that it can work in first questions, but our ambitions are so that it can be applied to whatever treatment we want to consider. So a patient comes in and we just analyze the data, whether it's digital pathology, clinical data, sequencing, really whatever we can look at, and then predict this patient would benefit from adjuvant chemotherapy, this patient would benefit from ovarian function suppression. Do you guys have any specific research, engineering topics that you're excited about?
Yeah, I'm excited about when we apply our tests to clinical trials to see like maybe we can identify new subgroups of patients who previously not known to benefit from specific lines of treatment. Maybe that opens doors to like new lines of treatment that can be developed. I don't know, what about you, Linus?
Yeah, similar to you. And also I think like if we end up finding certain groups of people that benefit highly from treatment or don't benefit from treatment that maybe go against conventional wisdom on the subject, that would be really interesting to me, especially if we, can really show this is true with a robust set of evaluations that we really trust, then I think that would be a really, really exciting thing.
Yeah, this brings me to two thoughts. One is that there are so many negative trials where you would expect that there is an effect, but again, even clinical trials, when the drug is introduced into standard of care, they often are based on very rough enrollment criteria of how big the tumor is. And we know that there is a signal, but the signal is too weak to meet statistical significance. And maybe if you can identify patients who actually benefit, you can make the negative trial positive, which is one way. And second, related to that is that right now, clinical guidelines are, again, based mostly on those super high-level things. Is the tumor ER positive, HER2 positive? Is it larger than 5 centimeters? Is the disease in lymph nodes? And overall, it is prognostic, of course, but if you look at who should get what kind of therapy is based on that decision tree, essentially. And if you end up in one node of that decision tree, that's the treatment you're going to get. And sometimes you won't even be considered for other therapies. So like you're saying, you can even be considered for new therapy options that might go against the traditional wisdom.
And I hope we don't sound like we're bashing clinical trials too much because there's a lot of utility in them. But I think that brings me to another thing that I'm really excited about is that we've evaluated our model's ability to essentially simulate clinical trials that it didn't see during training. We can simulate them to a pretty high degree of accuracy. So this means that you can sort of do clinical trials without actually doing them, which of course saves a lot of time, a lot of costs. And that's another really exciting thing about our model.
Guys, thanks so much for chatting about the test and TAL platform. And thanks everybody for chiming in and listening to us, and hope to talk to you soon. Thanks a lot.